Hello, welcome to week 12. Okay, we're going to be doing the reproductive system today. So the reproductive system, I guess you saved the best for last. You may know that I am a reproductive physiologist, so this is more of my area of expertise as far as more of a gamete physiologist. However, I did learn about all the male and female reproductive. So I think of myself as a cell biologist first, and then reproduction because of dealing with the sperm biochemistry and all that goes along with that. And so I find this topic, a lot of people don't know too much about this topic, even though they might think they know about sex. But as far as reproduction, how your body works, how these systems are, what the anatomy is, there's a lot to it. And the thing is that I went into this field because this field is very wide open because we haven't been studying this field as long. And we put it in the back of the book, and it's like this taboo subject. We can't talk about this, and that kind of thing. But really, I think it's very, very important, especially just keep separate from the sex part, just talking about your reproductive system, how it works, and the anatomy and physiology of that system. So we'll start off, we have male reproductive and then female reproductive. We'll start off with male reproductive. Fairly straightforward. I want you to take these notes right here on the male system, here. And this gives the pathway from start to finish. Now there's a lot of structures here, but what I like to do is, because I figure I'm more of an expert here, I think that I can push on this part here and have you learn a lot of stuff. I think you'll learn a lot more from me, and I like to emphasize this. I figure that you may not get this kind of education anywhere else. So we'll start off with the male reproductive system, where you have the pathway, and this is the pathway that you'd have for, let's say, for sperm to actually go all the way out through the body. The things that I've put boxes around, those are accessory organs that, accessory structure, accessory glands that would the sperm wouldn't necessarily go through. Like, it doesn't go through the seminal vesicle, it doesn't go through the bulbourethral gland. But the seminal vesicle, the prostate, the bulbourethral gland are structures that contribute to the pathway as far as contributing the volume to semen or ejaculate, in males. So we have that pathway, and then I'll pull out the male structures. So first we'll start off with the, here's our male structure here, where we start off with the testis. Okay? So we have the testes, and they are covered with a dense regular connective tissue. It's called the tunica alberginea, and then that's the surface covering of the testis. And then they sit in a little sac that's called the tunica vaginalis. Now, the tunica vaginalis was formerly part of the peritoneum. Your gonads, male and female gonads, start actually up in the back wall of your body cavity right here, and you'd have this piece of strip of mesoderm, this tissue, these cells. You'd have the adrenal cortex, you have the kidneys, you have the gonads, either sperm or testis, or gonads is testis or ovaries. The testis have to descend and go outside the body. The ovaries would stay internal. So the testes, they descend. They take a little bit of peritoneum with them, as they descend through the inguinal canal and inguinal ring. We'll get to those structures later. What we have here is it's all connected to the body. And so what we have is we have the testis, then we have a sac that's sitting right on top of the testis. It's called the epididymis. And then from the epididymis, we have this structure here. It's called the spermatic cord. The spermatic cord is going to contain all of the blood vessels. You see the arteries and veins. There are nerves. There are lymphatics. There's also this tube right here. See it? That's called the ductus deferens, or what's called the vas deferens. This is the tube that gets cut during a vasectomy for male sterilization. Okay? And so we have these structures here. The scrotum, what the testes would sit outside the body, below normal body temperature, about 36 C, about a degree C or 2 or so degrees Fahrenheit, lower than body temperature, so that that's the optimal temperature for spermatogenesis. Okay? But the temperature here needs to be adjusted, and so if it's cold, what happens is there's two muscles. There's one muscle, it's called the cremaster muscle. We see the cremaster muscle here. The cremaster muscle surrounds the cord, and then that would contract and elevate the testes. Okay? Then there's muscle in the scrotum, it's called the dartos. Dartos is in the scrotum, and that elevates the scrotum. So if it gets cold, then the cremaster and the dartos contract, pull. Cremaster pulls the testis up, the dartos pulls the scrotum up. If it's warm, then they relax so that the testes can descend further away from the body. All right, so now we go through the structures, okay, in the pathway. Okay. We have the testis. The testis has little lobules here, and you have all inside here are all these microscopic seminiferous tubules. Here's what the seminiferous tubules look like under the microscope. Okay. Hopefully we see it. There we go. Okay, so hopefully you can see this. Okay, what you're seeing is high magnification of the testis. We go a little bit lower magnification. Okay. Right here at what is it? This is 100x total magnification. What you see is a whole bunch of tubes. Okay. These are the seminiferous tubules. Okay, so we have all these seminiferous tubules here. Okay. And then get this in a little bit better focus. It's in focus on the microscope. There we go. So we have all these seminiferous tubules. Okay. When we learned about meiosis, we started off with spermatogonia and we worked our way all the way down to spermatozoa. What we were doing is we're basically looking through a section, okay, of the seminiferous tubules. The spermatogonia are on the edges. And as we work our way down, spermatocytes, spermatids, and then the sperm are collected here in the lumen of the tubule. Right there, the lumen of the tubule. And that's where the sperm is. Then the sperm, basically there's fluid produced. Okay? There are cells, they're called Sertoli cells. They are helping to mature the sperm. And then pushing, basically creating the fluid, and then the sperm are passively kind of washed out into the rete testis, it's kind of collecting area, and then out through efferent ductules, and then into the epididymis. Okay. What we see here is under high magnification. We see get a little brighter here. There. We can see here's one big tubule, okay? Here's another tubule, and here's another tubule. Okay? On the edges, the smaller cells, real dark stained and perfectly round with the real big nuclei, those are the spermatogonia. Okay? Those are your stem cells to make all the sperm. We have Sertoli cells, are these big ones here, and then we have spermatocytes, the spermatids here, going through spermiogenesis to make sperm. Okay? The cells on the outside, okay, you see them here, not in a tubule. These cells right here, okay, those are the interstitial cells of Leydig. So these are the Leydig cells here. These are the cells that are producing testosterone. You have two major cell types in the testis. You have the Sertoli cells, and you have the Leydig cells. Those are two important cells, and we'll talk about them more later after we talk about the female. But basically, the Sertoli cells are guiding spermatogenesis, and then the Leydig cells here are going to be guiding, basically producing testosterone. Okay, so we have testosterone production here, and then in here we have spermatogenesis. Now, when the sperm is collected, it's collected into structure. It's called the epididymis. The epididymis, I can show you another slide here. And I have to get this in focus real quick for you. Okay. I'm going to get it in focus on the seminiferous tubules. Okay. There we go. Here's the seminiferous tubules. Now it's a different stain color, but you can see here Here's an example of a tubule here. Okay. So then we have here, here's our tubule. Okay. Notice it's quite a few cell layers thick. And then we have some interstitial cells here. We got some interstitial cells here between tubules here. Okay. That is undergoing spermatogenesis in the testis, seminiferous tubules. We go over to the epididymis. Take a look at the cells here. Okay. You see that? Okay. Got some connective tissue here. Big lumen to store a lot of sperm. Okay. Simple columnar epithelium. Stereocilia, okay. Basically little microvilli that are going to help absorb some of the excess fluid, will help absorb extra sperm if it's not used, but it will keep the sperm there, okay, and kind of push the sperm along. And what happens is the sperm, actually, as they go through the epididymis, takes usually about 2-3 days, usually at least 2 days, to go through, and it starts off at an area of the epididymis that's called the caput. The caput, the head region. The sperm, it looks like mature sperm, but it doesn't swim. It doesn't have the capability to fertilize the egg. When we get into the cauda, or into the corpus, okay, is the central section of the body. And then we get to the end, it's called the cauda. Okay. So we have here, okay, here's the caput, and we have the corpus, and then right before it makes this turn here, that's the cauda. And the cauda, caudal epididymis, we take the sperm out of here. This sperm is totally functional, ready to go, can fertilize an egg. And so it gains functional maturity as it makes its way through the epididymis. If the sperm is not used, it gets reabsorbed. Okay? We have sperm here, and then sperm in the ductus deferens going all the way up. Okay? The ductus deferens, okay, or vas deferens, goes up through the spermatic cord, and then it goes through a structure, it's called the inguinal ring. So right here, this ring right here is the inguinal ring. It's the outer part, basically outside the body. Okay? And then you have, it's the outer border of what's called the inguinal canal. And we have our inguinal canal in here as it's making its way up in through the abdominal wall to go into the peritoneal cavity. It's going. And what happens is the ductus deferens goes up and around, okay, both sides, there's a left and right, up and around the bladder, goes back in, and it goes back near the prostate. Now, here's another structure, or another model showing you the structure. So we got testis, epididymis, ductus deferens, going to come in here through the inguinal canal, up and around the bladder. Right here is the ampulla of the ductus deferens. The ampulla of the ductus deferens is a wide part of the ductus deferens. It is going to be where the sperm that's ejaculated is going to come from. People assume that if they ejaculate, like the sperm would come from there because that's where they're stored, or maybe the epididymis. But actually what happens is there's sperm all the way in this tract. When ejaculation happens, this sperm here gets pushed into this duct right here. It's called the ejaculatory duct. Okay? And the ejaculatory duct, basically the sperm from here gets pushed into the ejaculatory duct, and then that sperm gets ejaculated out. Okay? What happens is you have basically, males would have the ampulla, then right here, this structure is the seminal vesicle. The sperm from the ampulla, the semen from the seminal vesicle, and semen secretion from the prostate gland. Okay, here's the prostate, okay, right there, number 7. There's the prostate. Okay, those secretions from here, here, and here get put into the ejaculatory duct. They put pressure on the prostate, and that's enough pressure to cause stretch receptors, to cause a reflex, spinal cord reflex, to cause peristalsis, and that would be the ejaculation reflex. So there's an emission reflex that's basically pushing the sperm down in here and the semen down in here. And then that causes the ejaculation reflex of muscular contraction to get the sperm and semen out. Now, the thing is that a lot of times men, they think like if they have a vasectomy that they would not be able to ejaculate. And actually that's not true because If a male, say, is going to have a vasectomy, they're going to cut here, sever this, so that male might be still making sperm, and sperm would all just get reabsorbed. Okay? There would be no more sperm in the tract, but that's only about, of ejaculate volume, sperm is only about 5 to 10%. So you'd still have secretions from the seminal vesicle and the prostate, and that makes up 90% of the ejaculate volume. So basically, the ejaculate volume would be negligible difference after vasectomy. Another area where you'd have some ejaculate would be right here. It's called the bulbourethral gland, right there. That's the bulbourethral gland in the pathway, basically. Okay. Going through here, the ampulla, then hooks in with the seminal vesicle to the ejaculatory duct, into the prostate, hooks into the prostatic urethra. Now with the ejaculatory reflex, you also get contraction of the internal urethral sphincter, so everything goes out instead of up. Okay, there. But then it would go from the prostatic urethra into this section from about here to here, where it enters the penis. This structure right here, this part of the urethra, is called the membranous urethra. It's when it's trying to get out through the perineum, okay, and then into the penis. All right, so now we've reached the penis, and basically I have finished the pathway up where the membranous urethra, bulbourethral gland, then we have the penile urethra, okay, and then the external urethral orifice here. Sometimes this is called the spongy urethra, because this part in the darker, deeper blue is called the corpus spongiosum. It completely surrounds the urethra. Sometimes called the cavernous urethra. I don't really like that term. Here's the corpora cavernosa, right here in the lighter. That doesn't actually touch the urethra, so I don't like that term. I like penile urethra or spongy urethra is fine. External urethral orifice is here, out where it leaves. Okay. So those are structures, and then it kind of just shows you the whole entire pathway. So we've completed the pathway for male reproduction. Okay. So that you've seen what's inside the testis. Okay. We have the structures here. Okay. And then now we can get into the penis. Okay. So now, penis, what does it look like? Well, basically, what you have is the penis. It has erectile tissues that fill with blood. And there are basically three bodies. There's two corpora cavernosa and one corpus spongiosum. Now, if I took this model, okay, take it off, we can see the structure here. We see three different bodies here, and then the urethra inside here. Okay. Well, basically what you have is this. Okay. In the penis, there are the two large, and if you go right through the body of the penis, or the shaft part, the two largest erectile tissues are called the corpora cavernosa. Okay. Those are the cavernous bodies. Here. And there's a left and right. Okay. The smaller one, okay, is the corpus spongiosum. Is this one right here, and it surrounds the urethra. A lot of people are surprised to see that the urethra actually goes through the ventral, or the inferior, closer to the inferior surface of the penis. It doesn't go right through the middle of the penis. It actually goes through the corpus spongiosum, which is actually on the ventral side. You have the corpora cavernosa is the ventral side and gives most of the size of the penis. And so here's the structure here of all the erectile tissues. All right. So under parasympathetic response, then those would vasodilate and you'd have filling with blood. Okay. On the penis, though, okay, we can see on this model here, it's pretty decent to show you, okay, structures. Okay. We can get a side view here instead of a cross section. Side view, we can see the corpora cavernosa. There's going to be two of them, left and right. Then there's the corpus spongiosum here. The cavernosa, okay, is going to go in, and when it goes into the body, it hooks in, and it's attached by ligaments, okay, that hold it in place. And this innermost part is called the crus, okay, right here. And so the innermost part is called the crus. There are two crura, because there's a left and right corpora cavernosa. Then there's the spongiosum. The spongiosum, on the distal end, okay, outermost, is enlarged. This is called the glands. Some people would call it the head. That's not a proper term. We'll call it the glands of the penis. And so the glands, or glans penis, is going to be an enlargement of the corpus spongiosum. Then that is covered by skin. It's called the prepuce. The prepuce, otherwise known as the foreskin. So if you've heard of head, that's glands, and foreskin, that is prepuce. So we'll use glands, prepuce. I want to use proper terminology throughout this male and female reproductive tract. So glands and prepuce here, it's enlargement of the corpus spongiosum. Then the corpus spongiosum is not very big compared to the cavernosa until it reaches inside the body, okay, internal, where it's connected to the body, held into the coccyx by ligaments. Okay, and this enlargement right here is called the bulb. Okay, so there's a glans and there's a bulb. The bulb, okay, right by the bulbourethral gland. The bulbourethral gland, they call it that because it's by the bulb and the urethra. The bulb and the two crura form the root of the penis, and that's holding it in place. As you notice here in this this model here, okay, a good amount of the penis, okay, here is actually internal, inside of the body. All right, so that shows you all the structures of the male. Okay, so now one last thing to talk about with male is going to be sperm. Okay. So the sperm, they're made. Right? What do they look like? A lot of people think they look like tadpoles or something like this. Here's the structure of human sperm. Okay? And a lot of sperm types look like this. This is very common among a lot of animals. Okay? And vertebrates, most vertebrates, even some invertebrates would have sperm that look like this. Okay? Sea urchin sperm look kind of like this. Their heads are a little bit smaller. Sea urchins have less DNA. Those are the animals that I study. Okay, bull sperm looks like this. Human sperm. This is a example here. What you have is actually the head region is not actually football shaped or round. It's actually shaped more like a paddle. So I'm showing the flat side of the paddle. Okay, something like this. But actually, when you turn it sideways, it kind of looks almost like a diamond shape, and it's real thin. So it looks flat and round on one side and then kind of diamond-shaped on the other side. In the sperm tracking software, when it's basically trying to measure how fast the sperm is swimming, it can lock onto the sperm because it sees it kind of shimmering as it's swimming because you see it flatter and then wider as it's swimming through. So there's the head. Then at the equator of the head and the distal end of the head, there's a cap on there. It's called the acrosome. The acrosome is actually a modified lysosome, so it contains digestive enzymes. Okay? And that would penetrate through the corona radiata and the zona pellucida of the egg to actually get inside the egg. All right, so now we have the acrosome, okay, has the digestive enzymes. We have the head, real tightly packed DNA. And then we have this neck region, or what's called the midpiece. The midpiece contains mitochondria. Okay? The mitochondria is going to be basically for aerobic respiration. So it's going to make ATP to drive the tail, okay, to swim. And so for the energy for swimming actually comes from mitochondria here, which means you need some kind of sugar, glucose, or fructose, okay, for nutrient. And then also it needs oxygen as well. So there's some oxygen in the fluid, fine enough, and oxygen would be in the female reproductive tract. In males, there is some fructose that would be in the semen. Okay, so there is a little bit there for the sperm. Okay. The way that semen is formed is basically, okay, and you can see this in your PowerPoint notes. Okay, that basically what happens is the majority of ejaculate is going to come from here, seminal vesicle, and here in the prostate. And these are kept separate because what you want to do is keep these, basically we have fibrinogen here and we have clotting factors here. So if we had them together, they would basically clot or clump or congeal, increase their viscosity. What you want is kind of the way that ejaculate works is like an epoxy, where an epoxy has two liquids, okay, two different tubes, and then you keep them separate, they're liquid. When you put them together, then they're going to form the glue and harden. Well, that's what happens with the ejaculate. When you mix the seminal vesicle fluid, seminal fluid, with the prostatic fluid, after it goes out, these two fluids get to mix, then that would cause clotting, that the fibrinogen would form a clot, and then that increases the viscosity in order to keep the ejaculate inside the female tract. Sits there for about 20 minutes, and after about 20 minutes, the prostatic fluid contains fibrinolysin, which breaks up the clot. So then the clot dissolves, gets thinner, and then the sperm can actually, when it starts being capacitated, it wakes up and it starts swimming. When it starts swimming in the female tract, it's actually going to be able to swim out of that clot because the clot is loosened up. Okay? So that's basically how it works. It's like an epoxy. Keep them separate so it's low viscosity to get out, higher viscosity to keep in the female tract, and then lower viscosity so that the sperm can swim up out of it. Okay, so that's basically how the structure works. All right. Last thing we have the tail that contains a flagellum. So we have 4 parts of the sperm. Make sure you know those 4 parts. All right. Okay, so that finishes up male reproductive physiology, and let's go into female reproductive phys. So in females, okay, first thing we want to talk about is the ovaries. Okay? Now, where are the ovaries located? The ovaries are in the, so you can see right in here, okay, in the abdominal, cavity. Say, almost pelvic to almost abdominal cavity here. Okay? And notice you see is a lot of times you see pictures of the female tract, and you see this head-on representation where you'd see the vagina going straight up to the uterus, and then the fallopian tubes going straight out to the ovary, and you'd think that it's all this straight shot. In reality, these are all at 90 degrees to each other. You have the vagina is at 90 degrees to the uterine cavity, and then the ovary, the fallopian tubes and the ovary is at 90 degrees to that. So it's a zigzag, actually, orientation to fit it all in there. Okay? And so this is actually, the actual orientation, that it's actually sitting like this, and then the uterus, the fundus of the uterus, top part right here, is sitting anteriorly tipped forward. Okay? So we have the structure here, and here's the ovary. Okay. Closer inspection of the ovary. Okay. Here's the ovary. Okay, here's a big ovary. I'm going to come back to this. We have another picture of the ovary, and I want to show you here. Okay. So I like to call this my uterus on a stick right here. And for this model here, we're kind of going to ignore that there's a 1-month embryo here. But what we can see here is that there are ligaments here. This ligament, this big flat ligament, covers everything up. It's called the broad ligament. Okay? And then the way it's sitting is it would be sitting here, sitting forward. Okay? Here's the fundus of the uterus. And then you have the round ligament. The round ligament is going to go forward, okay? Say anterior to the abdominal wall, right at the pubis. Okay? So the pubic bone and the abdominal wall, called the round ligament, because this ligament goes up, around, and comes back. It's almost like circular. Okay? There. So we have the broad ligament. Okay? Here you can see it best. We have the round ligament here. Then, okay, right here, if you can see it up close, we have this structure right here. That is the ovarian ligament. That is holding the ovary in place. Okay? The ovary is actually being held in place by ligaments. And then you have the fallopian tube, or what's called the uterine tube. The uterine tube is actually not touching the ovary. It's just being held in place right over it. So you'd have the ovary and then the fimbria, the fingers of the fallopian tube. My arm is a fallopian tube. Okay? The fingers are right here over it. They don't actually touch it. What happens is you actually have, here's the ovary, and then we have the fimbriae has cilia on it, and the cilia create a little bit of current to suck the oocyte when it's ovulated into the tube. All right, so now what we have to do is we have to grow the oocyte in a follicle. And I've talked about this before, maybe just like briefly hinted about it, that the ovary grows oocytes in follicles. These follicles have two cell types, just like how the testis has the Sertoli cells and the Leydig cells. The ovary will have the granulosa cells and the thecal cells. So we can see here on the model here, okay, on this one here is we have these tiny little oocytes and they're sitting in an egg nest. Okay. Those are called primordial follicles. Okay. You have a whole bunch of these. Young women would have a whole bunch of these, and then as you get older you have less and less of these. Okay. Every month you have several hundred of these decide that they want to become the egg that gets ovulated. So a lot of these become, and they grow, become a primary follicle. Now, a primary follicle, there's a secondary, tertiary, they don't follow oocyte, okay? They just call them follicles. So you would still have a primary, oocyte in a primary follicle, maybe even a primary oocyte in a secondary follicle. They're kind of independent, but you have a primary follicle here. The primary follicle here, if I can show you, okay? Actually this one's a real good one. We have the oocyte with, here's the nucleus, here's the cytoplasm. You see this white ring right there? Okay? That white ring right there, that is the zona pellucida. And then outside that, these gray cells with the blue nuclei, these are granulosa cells. With the pink cells on the outside, those are the thecal cells. The primary follicle has one layer of granulosa. Okay? We go up to secondary follicle, here, okay? Sometimes called maturing follicle. Secondary follicle has at least 2 layers of granulosa. Okay? 2, 3, 4, 5. What happens is these layers of granulosa, they start producing a fluid. It's called follicular fluid. And after a while, that fluid forms in a little pocket. And that pocket is called an antrum. Now this is a real late stage follicle here. This is a late tertiary follicle, so they call it a Graafian follicle. It has this huge, what we call antrum. But I'm going to try to find these under the microscope for you. So here are here's the rubric as far as following like what is in the follicle. Now remember, the oocyte is growing inside these follicles. The follicles are made up of granulosa cells and thecal cells. And so we see the primordial follicles, primary, secondary, tertiary, what they have. The tertiary follicle has this antrum. It's a pocket of follicular fluid. And we'll see this under the microscope. Then we have a Graafian follicle that has a really big antrum. And then we have ovulation. And at ovulation, that follicle, the oocyte shoots out of it. So we see it here where it's starting to get squirted out. And then what's left, right here, this is at ovulation. They call this a corpus hemorrhagicum because it's a hemorrhaging body. It's just ovulated. And then eventually this becomes the corpus luteum. The corpus luteum is called yellow body because you see it's yellow here. It has the yellow color. Corpus luteum starts making a whole bunch of estradiol and progesterone. This, if you're pregnant, is going to maintain the pregnancy. It maintains the first half of the, really this guides the first half of pregnancy. If you're not pregnant, if there isn't an embryo, what they call blastocyst to implant, and if that blastocyst isn't producing human chorionic gonadotropin, then that blastocyst is not going to tell the corpus luteum to persist. Okay? So, if you have a month that you're not pregnant, this is only here for about 7 to 10 days, and then if there's no hormones and there's no pregnancy, then this stops making the progesterone and estradiol. And then it turns this kind of white color, so they call it the corpus albicans, like albina, albicortuna. Okay? Albino, albicans is white, so this is the white body. So we went from the yellow body, making lots of hormones, to the white body, not as much. If you're pregnant, this stays around for about 4 or 5 months until the placenta can start making hormones. If you're not pregnant, this stops making as much estradiol and progesterone, and then a couple days later, then menses happen, so menstruation. All right, so here's the notes for all of this. Okay. Now let's see these under a microscope. Okay. It may take me a little while to find these. Okay. I'll do my best. Okay, and I'll tell you the magnifications on here. All right. We see here. It's a little brighter. Right there. Okay. Now, I can see here under the microscope. Okay. It's because I know what I'm looking for. So I can see things like secondary follicle. Here's a graphene follicle. Here's a bunch of primordial follicles sitting in an egg nest. Well, let's go up under higher magnification. Okay. That was at scan power, 40x total magnification. Okay. This is 100x magnification. At 100x, we see all of these little primordial follicles. See them all? Okay. All those bubbles right there, those are primordial follicles in an egg nest. Now let's find a primary follicle. Primary follicle is going to have one layer of granulosa. Okay. All right, so I just have to hunt around for these things. Try to find a good one for you. Here's a good one. Actually, I found two. Here. Let me get it in focus. Bring it up a little bit. Okay. Right there. Okay. These are primordial follicles. Right here, and right here. We see an oocyte. The nucleus, cytoplasm, we see this red ring. The red ring is the zona pellucida. It's a protective layer, protect the oocyte after ovulation. It's what the sperm has to burrow through in order to fertilize the egg. Okay. And then we see one here. Okay. Outside of this red ring, we see one layer of thick, fluffy cells. That's the granulosa cells. Granulosa cells have FSH receptor, a follicle stimulating hormone, and then they will stimulate the follicle. That follicle stimulating hormone will stimulate granulosa cells to grow and mature this oocyte. Outside of that, these cells out here, those are the thecal cells. They kind of have a little bit of a bluish tinge to them here. They're flatter. Thecal cells, you see them over here. Okay? They're kind of like the Leydig cells where they're producing hormones. So we have, just like in the male has one cell type that's intimate with the gamete trying to mature it. So you have the Sertoli cells or the granulosa cells. They both have FSH receptors. And then you have cells on the outside that are primarily interested in making hormones. And so on the outside, you have the Leydig cells making testosterone, or here, the thecal cells making estradiol, and then later they make progesterone inside the corpus luteum. So we'll see this. Okay. Just to give you a close-up, when the egg is actually ovulated, a couple of the granulosa cells go with it. And so these are formerly granulosa cells, and they call these now corona radiata cells after it's been ovulated. The white layer right in here, that's the zona pellucida, the cytoplasm nucleus there. Okay. All right, when this is ovulated, I wish they would have drawn this nucleus at metaphase 2. Would have been nice, but here's the model right there. Okay. So now let's go up to a secondary follicle. Found a nice one here. Let me get this in focus a little bit more for you. Okay. Right here. Okay. This is real nice. You can see the oocyte. See the zona pellucida, and I see 1, 2, 3, 4, 4.5 to 5 layers of granulosa outside of that good thick layer of thecal cells. Okay. This is a secondary follicle, sometimes called a maturing follicle. In here, I see little pockets of follicular fluid, but it hasn't kind of coalesced into a nice defined pocket, which we call an antrum. me try to find this in a tertiary follicle. Here's a tertiary follicle. And this is under scan power here. I'm sorry, under 100x power. So this is a lot bigger than the secondary one. The secondary was up here somewhere. Here's the secondary. So you can see the secondary here, the size of it. Before we were at 400x. Now we go over here, and here's a tertiary follicle here. This is a tertiary follicle. With the tertiary follicle, you can see the antrum right there, that pocket of follicular fluid. Okay? The granules the cells are producing. Okay? This will be the corona radiata. All right, then we go to the Graafian follicle. This is a biggie. Okay? Look how huge it is. It's huge. You could fit 100 primary follicles inside here. Huge antrum. This one will probably ovulate here. Okay, so those are your structures here. Okay, all right. So that covers the pathway, or I'm sorry, the progression of the follicle all the way through. And so after ovulation, then it leaves. Then what you have left is the corpus luteum. On here, I think there is a corpus luteum on this one, which means ovulation already happened. Okay? Yeah, here's the corpus luteum. It's huge. It's even bigger than the Graafian follicle. Here's the corpus luteum. Okay? This one, you might have several corpus lutea, and the reason why is because this one just has one. Although some of these might, ovulate, have ovulated. This is in a rat, so they have multiple in litter, versus in humans where you just have one per ovary that would ovulate. Okay? So in multiple, when animals that super ovulate have multiple eggs that are released, they would have multiple corpus luteum. Okay. All right. So now we go into the female tract. The females. I can turn this off. I think I've gone through most everything I wanted to. Oh, not yet. Okay. Got one more thing for you. The females. Okay. Right here, female tract. We're going to go through the pathway all the way through. So if you have an egg that ovulates, okay, say, go all the way through. Okay. If you have fertilization, it would get fertilized here in this fallopian tube here, okay, or uterine tube, and then implant in the uterus. If it doesn't, it usually dies, and then it'll degrade somewhere in the tube or in the uterine cavity. But if we go start to finish, let's say everything, where we go all the way to going out. Okay, pathway. It starts with the ovary. We go through. So let me go through the structures. Okay, here are the structures for the female, all the way from the ovary to what we call the vestibule. Okay, so start off. We have start here. Okay, we have the ovary, and then from the ovary we have the uterine tube. Now the uterine tube has a bunch of parts to it. Now they're not all equal sized parts. We just have the fingers right here. Those are the fimbriae, and then the part where the fingers are attached right in here, that's called the infundibulum. And then most of it right in here, all the way through, okay, to about here. Most of the uterine tube is what we call the ampulla. Ampulla is wide part. Okay? So it's the widest part, it's the longest part. It says that's where fertilization happens. Fertilization happens in the uterine tube, and it happens in the ampulla most likely because in the ampulla that's 90% of the tube, so that's where it's going to happen. Then it starts to narrow as it goes into the uterine cavity. And as it narrows, they call that narrowing, and that's an isthmus. Okay? and just like two landmasses connecting two landmasses where it's narrow, it's an isthmus. And the isthmus, sometimes they call it part of the uterus or sometimes part of the uterine tube. The uterine tube is technically part of the uterus, so it's all the same. And then that would go in. Now the ampulla, they say it's the wide part, and they show it on like all these books. They show it like it's this big wide tube, like you can drive a truck through there, and that's where the sperm swim up and everything. There's lots of nooks and crannies and crevices, and so it makes it more difficult for the sperm because a lot of the sperm would fall out of the tract. Sperm's got to swim through a tiny little hole. It's called the os to get into the cervical canal and then into the uterine cavity. It's got to pick one way or the other, and then it's got to go upstream, and it's got to go through the ampulla. Let me show you a cross-section of the fallopian tube. The assumption is that it's this wide open tube, and that's how they draw it, in all these textbooks, but in reality, it's not. Let me show you this. Okay? Right there. Okay? Let me get it in focus. You see that? This right here is the fallopian tube. This is the lumen of the fallopian tube. Do you see all these like nooks and crannies and crevices? Okay? There's lots of them in here. There's lots of folds and rugae all in here, okay, to make the job of the sperm more difficult. Plus, this is all ciliated. Okay? Remember that the egg is ovulated and it's moving along. It doesn't have a motor like the sperm. Okay? It's being passively pushed. So there's cilia pushing fluid through the tract. So it's kind of going downstream through here. And it's really tight in here. Okay? So it's not, really not not easy to navigate through. It's one thing I want to say that, for some people, if they have uncontrolled STD infection, it gets really, really bad. Like chlamydia or gonorrhea, that gets so bad to the point where it goes up into the fallopian tubes, it can cause sterilization. The reason why is because if you get inflammation and scarring in this tube here, it doesn't take much that you can completely scar over this tube and sever the tube, just totally scar off the tube, and then if it closes off completely on both sides, you've now basically given yourself a tubal. So you want to make sure that you don't, if you have STD, take care of it because this could seal up pretty easily. Now, one new procedure they're doing, rather than tubal ligation where they're cutting, they're doing, it's called the Escher procedure, where they actually do a dilation, put it up into the tube in the office. Okay? They can do this in the office now. They go up, and they put it into the tube, and it looks like a little pen spring with a little say Teflon bean that they basically, plastic bean, that they take that spring and they put it in there and they twist the spring and it scars up here, beats this up, and then it locks in place. And then as this is all destroyed from the spring twisting up in there, then it grows back over and completely seals over that bean and over the spring and closes everything up. So that's a new procedure. All right, so we've gotten to the uterus. I'm going to go to a different uterus on a stick just so you can see the structures. Here, this one's cut right here. So we've gone into the uterine cavity. Ignore that there's a 3-month fetus here. But from the uterine cavity, then we have here, this hole right there. That little hole is called the internal os, and then it goes through the cervical canal. This chunk right here, called the neck or the cervix. So right here, this is the cervix. We have the internal os, cervical canal, external os, this hole right here. Here's the external os, this tiny little hole right there. Okay? That hole is like that, okay? Except when you're about ready to deliver a child, you maybe have this hole is a couple centimeters wide, and when you go into labor, you have to wait till this hole gets to 10 centimeters wide, 4 inches, okay? For the baby's head to come out through here. All right, so in this structure in here, we can also see the fornix here. Okay? Fornix is the deepest part of the vagina. Okay, so that's the fornix. then we have the vagina here. So, we got fornix here. Okay, here's the uterine cavity to the. You can't really see the os in the cervical canal, but we do know that cervix is in this area right here. Kind of has a cone shaped, and it dips down into the vagina, and then it's surrounded. The deepest part of the vagina surrounds the cervix, called the fornix. Okay, then you have the vagina. Okay, females who have this intact, they would have a hymen, would be right here, as connective tissue covering. Okay, but typically you have the vagina, and then the orifice of the vagina opens up into an area. It's called the vestibule. The vestibule is mucous membrane, internal, where it has the opening for the urethra. So the urethra opens into the vestibule, and the vagina opens into the vestibule. On the lateral sides of the vestibule, then you'd have the labia minora, okay? And then lateral to the labia minora, you'd have the labia majora. Okay? Here's the glans clitoris. This is female erectile tissue right there. This is homologous to the corpus cavernosa of the penis. Okay? The corpus spongiosum, homologous to some erectile tissue that's in we call the bulb of the vestibule. So there is some erectile tissue inside of the vestibule, inside where the inside of the labia minor. All right. Going back to the structure, we've gone all the way through external os, vagina, vestibule, all the way out. The finish is female tract. Okay. In the uterine wall. Okay. The wall of the uterus has 3 layers. It has an endometrium. It is the innermost layer towards the cavity. That endometrium has 2 parts. There's a functional layer, what's called a decidual layer. It's the inner layer that's replaced every month, okay, during menses. So it is sloughed off during menses and then regrows. Okay. And there's a basal layer of the endometrium, and that endometrium is going to persist. We have the myometrium. That's most of the thickness, actually. Okay. That's the muscle, and then we have the parametrium. Is actually serous membrane, so it's actually a lot thinner than when I drew it. I just drew it thick enough so I could put the word parametrium there. It's the outermost layer. So in here, see it up close, we have the endometrium, myometrium, and then parametrium is this white line here. Okay. A couple other structures. We've covered most of the structures of the female. Okay. We have the mammary gland. Kind of just put this in here because it's kind of like after you reproduce. Okay. A couple things about the mammary gland. You have the nipple, okay, and then pigmented flat is areola. The bumps on there, okay, are called Montgomery glands, and then those are going to secrete oil after pregnancy in order to lubricate the nipple because the child would be suckling on it. Okay. In here, okay, how is milk production? How does this work? Okay. Here's the thing: there are lobes to this. This is a compound gland. Okay, multiple lobes. Okay, and everyone's different. It doesn't have to do with size of the breast. Size of the breast is basically how much fat is there. But glandular tissue, it's arranged in lobes, have anywhere from like 8 to 12 lobes, kind of radial, okay, radiate, like the hours of a clock. And right in here, what it's showing, okay, you can see is here is a lobe with 1, 2, 3, 4 lobules showing here that all go into this single duct. So we got 1, 2, 3, 4 lobules of this lobe. Here's another lobe that's going into this duct. Here's the 3rd lobe that's going into this duct. So you have all these lobes. They all end up in a lactiferous duct. So right here we can see parts of 3 lobes because I see 1, 2, 3 lactiferous ducts. Then, right underneath the areola, there's a wide part. It's kind of football shaped. Here's one, here's one, here's one. Okay? Those are lactiferous sinuses. They're right underneath the areola. Okay? And then they narrow again as they go up through the nipple. Okay? They don't merge. So that means if you have 12 lobes, you have 12 exits out the nipple. It's not like one exit, one shooting stream. Every lobe has its own separate exit out. Okay? they go through the lactiferous duct, lactiferous sinus, back into the duct. So the sinus is kind of a part of the duct that's underneath the areola. When the child is breastfeeding, it's not supposed to nipple feed because that clamps off the duct. Okay? The child's supposed to breastfeed, so you put as much of the breast inside the baby's mouth so when the baby latches on, clamps down onto the areola, and you want the baby to clamp down on the sinus. The baby clamps down on the duct, you don't get any milk, it closes it off. If the baby latches down on the sinus, then it opens up when the baby sucks because then it's pushing on the sinus, the wide part, and it can handle that. And then the milk would go out, multiple streams going out, and then the baby into the baby's mouth. Okay? So that's how it works. Okay, I know this is a long video. There's a lot to cover. I like to cover a lot, but we're kind of starting to finish it up. Now we have some, going over hormones and some endocrinology. So we've covered all the anatomy here. Here's the physiology of the menstrual cycle. Okay? So maybe I can hold this while I talk just so you can see the structure. What I've done is basically shown days, and this is a 28-day cycle. Now this is an average cycle with an ovulation on day 14. If you have a longer cycle, let's say 30 days, what you'd see is this first part, this follicular and proliferative phase, would be longer. So if you have a longer cycle, let's say 30 days, you'd see 16 days of this, 14 days of this. Okay? If you have a short cycle, like 26 days, you'd see 12 days of this, ovulation on day 12, and then 14 days of this. This is usually 14 days. This depends on how long your cycle is, and you usually ovulate 14 days before you menstruate. Okay? Now, the thing is that you ovulate here, 24 hours later, that's prime time for fertility, and you really have a short window post-ovulatory, like maybe max 6 to 30, but more like 12 to 24 hours post-ovulation for fertilization. But the thing is that the sperm can be ejaculated here 6 days before and still be in the tract. Okay? So a couple of things is like, you're not fertile for very long, but you are very fertile because the sperm can sit in the female tract for 6 days. Sperm can live, they're pretty durable. They can live in the male tract for 6 to 8 weeks. And the tract is full of it. So that means if you've had a vasectomy, you still have sperm in that tract. So that you would still be fertile for almost 2 months after a vasectomy until all the sperm is cleared and all the sperm would die off inside the male tract. Okay? Same thing as males would be fertile without ejaculating, so that's why the withdrawal method doesn't really work so well because there's still, if male has ejaculated any time in the last 6 months, there's still sperm in the tract somewhere. Okay? We wouldn't necessarily have to ejaculate, especially if the male has ejaculated recently. Okay? So there's still sperm in the tract. Okay? And the females can sit in the female tract for a good 6 days and wait for ovulation to happen. So you have to predict when ovulation is and then not to have had sex at least like 6 days before ovulation. So this is kind of, your window of fertility is usually a couple days pre-ovulatory and then a day post-ovulatory there. Okay. Plus, this is not always like on the dot day 14, quite variable, and the older you get, this gets uncoupled quite a bit that you could be ovulating almost any time. Even as you get older, your levels of FSH tend to go up, up, up. And what happens is, because estradiol goes down, FSH goes up, you might ovulate here and here. You could ovulate twice in the same cycle. Okay? And so then you can be fertile at any point in here. But normally, this is what would happen to someone who's young, female, who has a normal cycle, 28-day cycle. You start off, first day is menses. Okay? Menses, menstruation, okay? However, 5 days. Then after that, so this is what's happening in the uterus. In the uterus, there's menses days here, and then after that, then sloughs off the deciduous part of the endometrium, and then it has to grow it back. It's called the proliferative phase. So we're growing back the endometrium. There's ovulation. After ovulation, now the endometrium is preparing for implantation. So it's a secretory phase. You have these spiral glands that are going to be secreting mucus in the tract and basically making the making the uterus hospitable for the blastocyst to implant. So that's what's going on in the uterus. Okay? So pre-ovulatory we have menses, and the proliferative phase, and post-ovulatory we have the secretory phase. The ovary. Okay? What's going on in the ovary? We're growing follicles for the first part until ovulation, follicular phase. So these are our primary, secondary, tertiary, Graafian, and ovulation. Then afterwards we have the luteal phase. We'd have the corpus luteum all the way through making lots of hormones if you're pregnant. If you're not pregnant, then right about here, premenstrual, then you're going to see a decline in estradiol and progesterone. Okay? So I'm kind of covering the hormones here. Decline in progesterone and estradiol here during the end of it. And when those levels go down, 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 then it causes the uterus to undergo menses. So, what's happening with the hormones? This is not actual, this is just kind of relative amounts of these different hormones. And I kind of just spread them out so you can see them. I just staggered them here. So it's not like you have the most progesterone. It's basically just staggering, just showing relative amounts. What I want you to know is that you have FSH and LH. Those are the hormones, luteinizing hormone, follicle-stimulating hormone from the pituitary gland. Those hormones from your pituitary gland are going to go up. They're in a positive feedback loop with estradiol and with the follicle. So as the follicles get bigger, you're going to see more FSH, more LH, more estradiol, and when you get this spike, FSH, LH, estradiol, you're going to get ovulation. Then afterwards, it goes back down, you'd have the corpus luteum. The corpus luteum produces estradiol, we call it E2, and progesterone. The corpus luteum, if you're not pregnant, then it becomes corpus albicans, those levels drop off. If you're pregnant, these actually stay up nice and high. Okay? They keep going up. So this is what I want you to know. I want you to know that during ovulation, you see spikes in FSH, LH, and estradiol. Post-ovulatory, you see spikes in estradiol and progesterone. Now, when taking oral contraceptives, hormonal birth control, the pill, okay, the most common pill is a combination of progesterone and estradiol. Usually an estradiol or a progesterone analog like norgestrone or something. Okay? Some people have heard the pill makes your body think that you're pregnant. Well, what I like to say is the pill makes your body think that it might be pregnant. Because every month your body thinks it might be pregnant with a corpus luteum and it produces estradiol and progesterone. So what happens is basically you're taking the pill, you are mimicking the luteal phase, okay, of your cycle. And so instead of going through a normal cycle and having increases in FSH, LH, this spike, especially in LH, causes ovulation. Okay? When you get these spikes, you ovulate. If you have estradiol and progesterone at the same time, it is a negative feedback loop that keeps LH and FSH low so you don't ovulate. What you do is the pill, you'd keep this low, or like keep this high, take these hormones, keep this high, keep these low, do this for 3 weeks. Then go off of it and go to this for a week. And then you jump this part so you don't grow the follicles, and then you do this for 3 weeks. Okay? And then you jump over here for a week. If you're on some other like seasonal birth control like Depo-Provera, you're basically doing a whole bunch of progesterone to keep LH and FSH low. You don't ovulate for 3 months, and you're just staying high here, high levels of estradiol and progesterone for 3 months, and then you go off of it, okay? And then you start up again. Okay? So this is the cycle. So I like to say it makes your body think that you might be pregnant for 3 weeks, and then all of a sudden then it goes off, and then you see the estradiol and progesterone drop, and then your body says, nope, I'm not pregnant, okay, menses, and you start the cycle. Okay? So this is basically how this works. Okay, the very last thing I want to show you, okay, let's talk about males and females, how we're not that much different. So I want you to take this, you can take a screenshot or draw this down, okay? And what it's showing you is that you have hypothalamus, pituitary, and your gonads involved. Okay? And you have this axis here. So in males and females, the hypothalamus produces gonadotropin-releasing hormone that causes your pituitary to release LH and FSH. LH and FSH have specific targets in the gonads. So the thecal cells are a lot like the Leydig cells, the granulosa cells, a lot like the Sertoli cells. Okay. So, the thecal cells and Leydig cells, they're on the outside, and they're responsible for making the hormones, estradiol, progesterone, testosterone. Okay. Then you have the granulosa and Sertoli cells, they're responsible more for maturing the gametes. Okay. Here, FSH, follicle stimulating the follicle. Okay. They're there. And there's a negative feedback loop. Now, in males, it's pretty straightforward. Testosterone has a short and long loop where it inhibits LH and FSH, inhibits GnRH, so to keep this in homeostasis. Okay? Keep these levels fairly constant. Okay? Sertoli cells make inhibin to kind of do this too, so they have some input on the negative feedback. So it just keeps these levels fairly constant. Males are constantly fertile. Okay? There are some daily changes where males would have a little higher testosterone after midnight, early morning hours, and then lower in mid-afternoon, daytime hours. Okay? In females, okay, sometimes it's positive feedback if there's just estradiol. That's in the proliferative or the follicular phase. If you have both, okay, estradiol and progesterone, like the luteal phase, then it's a negative feedback. So it switches between positive feedback in the first half of the cycle, negative feedback in the second half of the cycle. Okay? All right, thank you very much. Have a good week, and we'll see you next week.